Okay guys, we're moving on to the next part of the topic. So we discussed some of these at length, so I'll go over them fairly quickly, but hopefully you have a good idea, you have some examples up your sleeve. I'm going to give you some more examples that we didn't go over in too much detail, that you may want to draw upon it as well. So as I say the whole way through these videos, you can't have too many examples. So improvements in biotechnology have led to strategies that allow farmers to have more control over the genetics of their animals and thereby improve the quality of the product. For example, beef. One such method is the bisection of embryos, which involves splitting a single embryo into two genetically identical clone embryos. Using this technique, a farmer can increase the number of embryos with desirable characteristics, such as a tendency to marble or put on muscle from a valuable donor. The farmer can then implant these embryos into recipient cows, embryo transfer, to produce more cattle with the desired characteristics and thus improve the quality of his or her product. However, biotechnology is not completely foolproof. The pregnancy rate for each half embryo is 50%, with this number decreasing after freezing and even more so with more than two divisions. For each half embryo, only 25% result in identical twins. There are also very few practitioners in Australia that have the necessary training to carry out this method, and the procedure itself is time consuming, taking 20 to 30 minutes, potentially disrupting workflow. Overall, biotechnology has allowed farmers to improve the quality of his or her product through genetic management strategies. However, these methods are time consuming and still new and improving. So I've taken these straight out of the Master Ag Notes, which is just another example for us. Some of the examples that you'll be more familiar with is the ones from the book. So we looked at cows producing milk with a high lysine content. So cows have been developed in China that secretes milk containing lysozyme, an amino acid found in human breast milk that boosts the immunity of breastfed babies. The milk could potentially be used as a substitute for human milk and make milk formula. The researchers inserted human lysosomes into the nuclei of cells in the bodies of cows and then used a technique called somatic cell nuclear transfer, the Dolly Methods. Remember I showed you Dolly the sheep, which is Scottish sheep that was cloned, to clone the cows. The benefits of this is you're manufacturing a commodity with few replacements. There's obvious health benefits if you cannot breastfeed your baby and you want to have lysozyme in there. And there's a practical solution to an existing problem. So there is a problem with breastfed babies not getting the appropriate milk. Possible risks: unknown long-term impacts on human health, unknown long-term impacts on ecology through crossbreeding, so if the genes get into the population, loss of lysozyme through processing and pasteurization of milk, so you will lose quite a lot of the desired characteristic when you pasteurize the milk, unknown commercial viability, how much does it cost to create, and are you actually going to have money to play with at the end of it, so your cost of production, what does that look like? You should have a note of this if you're in my class. If you don't, please write this down again. Even if you do have it, maybe write it down again, or just highlight the key areas. Okay, this is an amazing example to have up your sleeve. Our other example that we used, EnviroPig. EnviroPigs have genetically modified salivary glands, which help them digest phosphorus in feedstuffs and reduce phosphorus pollution in the environment. Phosphorus is crucial for healthy growth in pigs. Unfortunately, 50 to 70% of phosphorus in grain is in the form of phytic acid, a compound indigestible by pigs. To overcome this, farmers traditionally add expensive supplements. So that's kind of the problem. The solution then, the transgenic EnviroPig synthesizes phytase in its salivary glands, eliminating the need for additional supplements or enzymes in the feed. By digesting more phosphorus, the EnviroPig also produces less phosphorus in its waste. So what are the benefits of that? You have increased efficiency of production in feed conversion growth rates, reduce cost to producer because you're not having to supplement it with additional phosphorus, and reduces environmental impacts and degradation because there's less phosphorus going into the environment. And again, the cons are the unknown long-term impacts on human health, unknown long-term impacts on ecology if the pigs outbreed and escape captivity, and what happens when those genes go into the environment, and unknown commercial viability. Can you make money from this? Okay, so hopefully this is all in there. And just another one that we looked at was the Aqua advantage salmon. So there's a transgenic salmon species that has been engineered to grow faster than traditional salmon. A growth hormone regulating gene from a Pacific Chinook salmon, with a promoter from an ocean pout was added to the Atlantic salmon's 40,000 genes. This gene enables it to grow year-round instead of only during spring and summer. The purpose of the modifications is to increase the speed at which the fish grows without affecting its ultimate size or other qualities. The fish grows to market size in 16 to 18 months rather than 3 years. So the obvious benefits is increased production efficiency. Okay, it's taking much less time to get it to market size. Allows for the conservation of wild fish populations. So they're arguing if you can grow this guy, you don't need to go out and fish for wild salmon. Reduce carbon emissions. Fish produced close to manufacturing and consumers, so they can produce them closer than you don't have to ship them. Basically, you can grow them in the same area. Reduce environmental impacts and degradations through controlling water and effluent usage. And commercially viable. So Aqua Advantage salmon is sold in the US and Canada, so it is making money at the moment. And again, negatives or potential negatives. Unknown long-term impacts on human health if you eat this fish, and unknown long-term impacts on ecology and natural ecosystems if species escape captivity. Now, I've spoke to you in the past about the aquaculture in Scotland, that these escapes do happen, and when they do happen, it can have a really adverse effect on the whale population in terms of diluting the gene pool. But also when you have these big pens, usually in the coast, we talked about sea lice, and the problem with sea lice is the farmed fish get treated and the sea lice are killed off, but the whale populations that swim past get infected and they obviously don't get treated with any chemicals. So there's some issues there as well that we've talked about in the past you may have notes on. The last thing then is Bogart Cotton that I'll discuss. There is other examples, but these are the main ones we looked at. Insect-resistant transgenic cotton variety developed by Monsanto. The variety has 3 bacterial proteins added. Each have unique mode of actions to kill the Helicoverpa bollworm larvae. The addition of the third protein increases the sustainability of the technology, as it becomes increasingly difficult for the Helicoverpa species to develop resistant to any of the Bacillus thuringiensis or the Bt proteins. The cotton variety itself is resistant to herbicides, glyphosate, glufosinate and dicamba, so we've already seen this in these videos, allowing for flexibility of herbicide use for weed control. The benefits then: increased production efficiency, reduced need for chemical pesticide application, which has financial and environmental benefits, allows for interruption of the pest life cycle, but there's quite a lot of negatives. Unknown long-term impacts on human health, unknown long-term impacts on ecology and natural ecosystem if the species escapes or spreads beyond where it's supposed to be planted. Potential for development of pesticide-resistant superbugs. What happens when you eventually will get one of the pests that can resist all of the proteins that are being released? That's not good at all. Or the production of a superweed, one that is herbicide-resistant. If the cross-family breeding occurred from cotton plants. So if this plant bred and produced a hybrid with a weed, that weed would have all of these desirable traits, but obviously none of the benefits. We don't want to grow weeds. So that would be a massive problem, trying to get rid of that weed. Okay, there's a whole bunch of other examples of biotics that I'll maybe come back to. I'll just pause this video at the moment. I'll come back to these in 2 seconds. Okay, so if you want to go back, make sure that you have all these examples up your sleeve. I gave you another example there with the embryo splitting. The more examples you have, the more you have to talk about and write about in the exam.